Hello, um, welcome to my channel. Today we're going to talk about DNA and mutation and what its effects are, what it can cause, and how can they be fatal if you inherit a defective DNA from your parents. In last lecture, we talked about DNA and how you inherit from your parents, how inheritance is passed down the generation from one parent to the child. And we discussed about, if you can remember, that in 1953, James Watson and Francis Crick used the electron microscope at Cambridge University at the MRC department to examine the structure of DNA. And this was what they found. It was in a spiral form, a ladder with a backbone of phosphates and sugars, with the rungs lined up with base pairs, which are adenine, thymine, guanine, and cytosine. These are the four base pairs that made up the DNA. But they are arranged in a way that way is called the sequence of that living thing. Every living thing has a way in which its DNA is sequenced, that is, its DNA is arranged. And you can see it here, and the base pairs are listed here: adenine, thymine, cytosine, guanine, and it has a phosphate, which is the sugars, the phosphate backbone. That is where the handles of the ladder is, kind of. Now, if this is arranged in a different way other than this way, because it should be arranged always in this way. Adenine should always go with thymine, and guanine should always go with cytosine. That is how it should be always. But if it is changed in any way, then that changes is called a mutation. That gene has mutated, it has changed. It is the arrangement of the molecules, these DNA molecules, that codes it codes for the amino acids that are used to code for the proteins that makes your body. So if the arrangement is wrong, then it will code for a wrong protein, and therefore it will affect the living organism on which the protein is. So you can see here the definition: a gene mutation is a permanent alteration in the DNA sequence that makes up a gene, such that the sequence differs from what is found in most organisms. So you will be different completely if you have a mutated gene. That is why it's not good. And that is what we are going to discuss today. Now, let's see. The next is this is the chromosomes, the sets of chromosomes that we have, the male and female sets of chromosomes. This part is the X, where the XY chromosome is. That is the male part. We all have 46 chromosomes, but 23 from our mom, 23 from our dad. So we have 23 pairs, which is 46 chromosomes. For the male, you have X and Y for the 23rd chromosome. You can see they are listed 1, 2, 3, 4, 5, up to 23. Number 22 and then number 23 is the sex chromosome. This is the chromosome that determines whether you're going to be a boy or a girl. I'll be talking about sex determination in our next lesson. But for now, we just know that for male, you have to have a Y, X, and a Y chromosome. For female, you have to have also the same 23 sets of chromosomes, which is 23 pairs, 46 chromosomes. But at the last set, which is the 23rd set, there are X chromosomes, no Y. There is no Y in a female. Females got only X chromosomes. So we will be referring to these chromosomes as we go, because this is where the defective genes are located, because it's inside these chromosomes that where the DNA is. And DNA, when it replicates, when it is copying itself, that is when mistakes are made. And those errors are called endogenous errors. They are endogenous, that is, they occur inside the gene. They are not exogenous. They are not outside the gene. So we will be discussing that, and we will be showing which chromosomes can get defective and the disease that they can cause that could be passed over to the next generation. Right. So you can see here, I'm showing how a DNA copies itself. This is one whole DNA here, but when it copies, it unwinds and divides into two, and each part will make a copy of itself. As it unwinds, it copies itself and become at the end of the unwinding, you have two separate DNAs, but they are the same. The copy should be the same. If the copy is different, then there is a mutation. And here I have illustrated a mutation here. Let's look at these four base pairs. The first one is, you can see, is red and green on this copy. This is the original copy, and this is the correct copy. And now let's look at the original copy here. You have yellow and red, you have violet and green, you have here yellow and red, and you have green and violet. And it copies itself correctly here. But here, there is a problem. If you look at the top, because this copy should be the same as this copy here. You have yellow and green. Yes, you have yellow and green here. But here you have yellow and red. Also you have yellow and red, which is good. Here you have green and violet. Here you have green and violet, which is good. But at this point here, there is a problem. Here you have violet and green, but at this point, which should have been the same as violet and green, now it is red and green. So there is a mutation here. The red one should not have been there. So this gene is going to have it's going to code for a different protein. So this is called a point mutation. One point is changed in the DNA. And once that point is changed, then you have a changed DNA, which will produce a wrong protein. So this is how the errors are made. And this can cause problems, which you will see as we go. Now here, you can see that these, as they are endogenous, they happen inside of us. And it's during the copying process that is when these mistakes are made. In humans, you can see approximately 10 to the 7, which is about 10 million cells, divide per second. Every second, there is 10 million cells in our body dividing. We have a total of 6 trillion cells in our bodies. But in every second, you can have an estimated amount of 10 million cells dividing. That is why we change from young to old, from old to death. So, because the body keeps changing all the time. What your body was consisting of yesterday is not what it consists of today. So, because of the cells that were in your body yesterday have died, and new cells were born in the copying process, and you are consisting of these new cells today. And the new cells you have today also are consistently copying themselves to new cells. The old cells are dying. So your body is continuously changing. What you were even in the last minute, you are not that right now. So this is how life works. It consists of life and death always taking place. Right. That is what it's called endogenous mutation. That is the changes that happens inside our body can be due to the massive amount of cell copying that is happening. And in that process, mistakes can be made within the copying process, and leaving us with mutated genes. And here again, we are explaining the point mutation as we have just shown in the DNA unwinding in the first picture. At one point, there is a change. Here you can see the original sequence is TAAC, TGC, AGGT. Okay, but here you have TAA, which is all correct, C, which is all correct, but where the T is, has a mutation there, because instead of having a T, it has a C. So this is a point mutation. This gene is not going to make the correct protein compared to what this protein, this gene is going to make. And I will try to explain that further later. You will understand what I mean. And the causes could make is that a point mutation, for example, can cause sickle cell anemia in chromosome number 7. Chromosome number 11. If you go back, you will see, if you pause the video and rewind a little bit and go back to the sets of chromosomes I was showing you earlier on, that we have 23 pairs of chromosomes. You can see where chromosome 11 is, and that is where sickle cell anemia and sickle cell disease can happen. If you inherit a bad copy that makes hemoglobin is the molecule that carries oxygen in our bodies. It is the molecule of blood that carries oxygen in our body. It goes everywhere in our body. This is why anywhere you punch in your body, there is blood coming out, because every part of your body needs that oxygen, and it is carried by the hemoglobin molecule. Hemoglobin is the smallest molecule in our body. It's the smallest cell in our body. It is so small that it doesn't even have a cell nucleus where it could have a DNA in. It doesn't have space even to carry DNA. All the space that hemoglobin have is used to carry oxygen and to transport it across the body, everywhere around the body. So now that begs the question: how do they take DNA from blood? If blood doesn't have DNA, if the red cells does not have DNA in them, they don't have even a cell nucleus in them to carry DNA. How do they take DNA from there? They take it from the white blood cells, not from the red blood cells. Okay, so that is what hemoglobin does. And if you have a bad copy of a gene that makes a bad hemoglobin, then you can have sickle cell. It can also give you a disease called hemophilia. Hemophilia is a disease that doesn't allow you to stop bleeding. If you cut yourself, you continue to bleed for a long time. Maybe sometimes you can even need medical help to stop the bleeding. Otherwise, you will just continue to bleed. You don't have blood clotting agents in your blood to stop you from bleeding. So that is also another illness. Both of them can happen in black people and in white people. Sickle cell also can happen in white people, but it's predominantly happening in black people. And hemophilia can happen in both races as well, but it's predominantly happening in white people. And they are all caused by point mutation, one mutation in the gene in chromosome 11, and then you can pass it to your child. If the parents are carriers of this bad copy, if you inherit a bad copy from your father and a bad copy from your mother, then definitely you are inheriting a copy of blood cell that cannot make the correct blood cell. So therefore, you have sickle cell or you have hemophilia. Okay, that is how that happens. Now, the next two types of mutations are categorized as frame shift mutation. And what frame shift mutation is, for example, uh, DNA, as I said, makes the codes for the protein. It codes for the proteins, but because it codes for the amino acids, and the amino acids code for the proteins that makes up your cells and your tissues in your body. So let's see, for example, um, in every gene has 64 codons, because it's the codons that make the codes. And the codons are always three sets of the molecules, of the DNA molecules, the A, C, G's, and T's. You have three of them forming one codon, another three forming another codon, you have another three forming another codon. You have 64 codons, almost 64 codons in every gene. And your body uses 60, 63 of them, and then one of the 64s are used to stop the coding of the protein. But the rest codes for the protein. But you can see that because of its tree, I've shown an example here in a sentence. If this was a gene that is supposed to code for a protein, and now it is expressed in a sentence, like for example, the fat cat sat, you can see that is a correct sentence. The fat cat sat. But if you have a point mutation, that this one is deleted, the T in the D is deleted, and you need three molecules to make a codon. So the He will take the molecule one from the fat, it take the F and bring it here to add it there. And then it will leave the AT alone without the third one. So the AT will take the C, and then it will be AT and C. And then it will leave an AT there. It will take the S, and you have AT and S. And then the last two, AT, they don't have a third partner. So therefore, this codon is missing one DNA molecule, set of molecule. So it will be wrong. It will be creating a wrong protein. Now look at it here. If the T is removed, instead of D, you have HEF, because it's taking the F and add it to the HE. HEF at. What's that? The body doesn't understand this. The gene doesn't understand this. It becomes confused, and then therefore it makes a wrong protein, as this is a wrong sentence. Nobody understands. It's just gibberish. So that is what a frame shift deletion mutation can cause. Right. Now you can see what the problems are here. Deletion mutations are actually the cause for a large number of genetic diseases, such as 2/3 of cystic fibrosis and what they call Cat-Cry syndrome. Cat-Cry syndrome is known as cri du chat syndrome. It happens in the short arm of chromosome number 5. You can see here, I'm showing you chromosomes number 5. The short arm here has a lot of genes deleted here in this area. So a lot of information is taken out. So therefore, that causes serious problems. Any child that inherits this gene will have this syndrome. You can have what they call cri du chat syndrome, which is known as cat-cry syndrome. That is, when the baby cries, the sound of the crying looks like cat is crying, like meow, meow, something like that. So that is called a cri du chat syndrome. The cystic fibrosis is also a life-threatening chronic disease, chronic condition, and it preliminary affects the lungs and the digestive system. It is a very serious condition, and it is a life-threatening condition. It is, for example, you can have up to 40,000 children and adults in America having this cystic fibrosis disease, and you can have about 105,000 children and adults in over 94 countries in the world, and you have over 10 million people carrying the disease in the world. Now, if they're carrying this wrong disease in their body, if they're carrying this wrong gene, they have a deleted gene in their genome, and they're carrying it, it's not affecting them. You cannot see it in them. They are not manifesting the illness. Well, they are within us. It's maybe me, I have it, I'm carrying it, and I'm not manifesting it. Maybe it's you have it, you are not manifesting it. So if you are a carrier, and you marry somebody else who is a carrier, then definitely you are passing wrong gene to your daughter or your child or your son, and then they could be cystic fibrosis. They have a cystic fibrosis condition, and that is a life-threatening situation where you're going to put that child really in a life-threatening condition throughout, and you, the parents, also will also suffer to look after that child. The problem is, we don't test ourselves before we get married. We don't check who we marry. We just meet up at a certain place, we like each other, and then we engage, we get married. And then in that marriage, we might have a child. and then the child, if we are both carriers of this bad copy of a gene, then we pass it to our child. The cystic fibrosis wrong gene is in chromosome number 7. You can go back into the chromosome picture I have shown you and see where chromosome 7 is. And cri chart, which is the cat cry syndrome, is located in chromosome number 5 in the short arm of chromosome number 5. And this is caused by a deletion. Now, a deletion is a frame shift, as I said. Like if this gene, if this molecule, which is this T, was supposed to be here, as this is the original one, you have TAACT. Now you have TAAC and the T is deleted. So it is going to shift the frame to the left because you need 3 codons. You need 3 base pairs to make a codon. So you have 1, 2, 3. That's TAA will be 1 codon, and C, it should have been C, T, and G to make another codon. But because the T is out of it, then you have C, G, and it will take the C. So it will shift the frame to the left. And then that is how a missing base pair will be missing at the end, because there is one missing here, and that will create the gibberish that we have just looked at in that sentence in the last picture. So this gene is going to be very wrong, and this is what creates these diseases, and you can pass it over to your child. So it's good to know who we marry. Sometimes it is good to test ourselves, but normally we don't do it. So, but these are the conditions, these are the risks that we have in front of us. Right. Now, this one is another mutation, which is also another frame shift mutation. Is called an insertion mutation. It is like during the copying process, a base pair is inserted into the gene. And you can see here the original sequence was TAACTG CAGGT. Now, but here there is something inserted. One C is inserted here, which is a cytosine. So you have thymine, adenine, cytosine, cytosine. Instead it should have been cytosine, thymine, and guanine, like it is here, cytosine, thymine, and guanine. But because of the C is inserted here, now it shifts the frame towards the right. Like the other one, the deleted one, shift the frame towards the left. So therefore, it's also going to complete with a gibberish. The gene will not understand what it is supposed to create. What the protein it is supposed to create is going to be a messed up protein, and therefore that could cause these following diseases. A Huntington disease is a condition that stops parts of the brain working properly over time. It gets gradually worse over time and is usually fatal after a period of up to 20 years. Huntington disease is an autosomal dominant disorder caused by the elongation of CAG, which is the cytosine, adenine, and guanine repeats. They repeat themselves and making the gene longer than it should have been, and therefore it is found in chromosome number 4 at point location number 16. So these are the problems that can happen during the copying process of the gene. And we carry them, we pass them on with devastating effects. So still yet, this is the consequences that we reap from getting married without knowing ourselves, without knowing our partners as well. We just get married and then we deliver a child. If we have bad copy of genes in our bodies, definitely we are going to affect our child. Right. Okay, now the next one is an inversion mutation. The inversion mutation is where the gene, the molecules, which are they will invert themselves. Instead of you have CGT, you have TGC. And this also happens in the copying process. So the inversion in a human genome can cause a number of diseases as well, including it can also cause hemophilia, and it can also cause X-linked disease. The X-linked disease is something that we're going to discuss next in this program, and is a disease that is found in the X chromosome of a woman. Um, the male can only pass Y chromosome. The female only pass X chromosome. For you to have a boy, a baby boy, the male should pass you a Y chromosome, and that goes to find the X chromosome of the mother, and then you have a boy. So the child will have X and a Y chromosome, making him a boy. If you have to have um, a baby girl, um, then the male should be able to pass you, as the woman, an X chromosome, and that goes to find the woman's X chromosome. So the X from the father and the X from the mother gives the baby two X's. So it will be XX chromosome. That makes the baby a girl. So this is how you become a boy and a girl. But we're going to discuss that further in the lesson, where we're going to have a big discussion on sex determination in humans and in animals and in plants. How do we have sex differences? We will discuss that. But for now, this is just a brief explanation of what the X-linked diseases are. They are diseases that came from the X chromosome, which is passed down by the mother, not the father. Okay. Here also you have substitution mutation. A substitution mutation also is a mutation where the nucleotides, these are the nucleotides, are substituted with another set of nucleotides. Sometimes the substitution can come and perform the same functions as what it has substituted itself from. So then the gene is happy, is going to produce the same thing that should have been there anyway, because what is substituted is producing the same thing that it has substituted itself from. But sometimes the substitution can be different. The substituted molecules can be different, and therefore the gene will produce a wrong protein again. And again, that can cause sickle cell anemia. And these also are carried by parents, and they are passed down the generation. Okay, now let us discuss what X-link inheritance is. Just to show you this illustration here. There's a father, there's a mother. The father is an affected father because the father, as a man, it has the X and a Y chromosome. That's the pairs of chromosome that the man has to be a man. Okay, so but the father has a mutated gene in the X part of it. So his mother may have passed him a bad X chromosome, a bad copy, and then he's got a bad X chromosome. But his father passed him a good Y chromosome, and he's got a good Y chromosome. So if he marries a woman here, the woman has two normal genes. Both the X, because the woman has XX, the man has X and Y. So the woman, if he's got two X's and they are all good, they get married, and these are the effects that could happen when they have a child. If they have a daughter, we should have two X's, because a girl should have XX, not any Y in a girl. So it will take an X from the father, because the father has given the mother an X, finding one of the X's of the mother, but the mother has got two good X's. So then any X that is chosen is a good one, but the X of the father is not a good one. So the baby will have a one bad X and one good X. So the baby is affected. But it will not show any symptoms. The baby will carry whatever disease this is going to be. So this is what a carrier is. You don't have two bad copies, you have one bad copy. So you are carrying it. This X will do the same job that this X should have done, but because this X is default, is a mutated one, this good one will say, okay, all right, just stay there, I will take over from you and I will do the job that the X is supposed to do. So this baby will have a good functioning X, but it's carrying a bad one. Now, if they have a son here, the son is unaffected because the son for you to be a son, you have to be a man and you have to have X and Y chromosomes. So you take a Y chromosome from your father, which is a good one here, and you take one X chromosome from your mother. Both X's, any one that you take is fine because they are all okay. So you have both good chromosomes. So you are unaffected. The son is unaffected. He is cool. He's okay. Now, in another situation, in another circumstances, um, in this scenario, the father was affected, but in this scenario, the father is unaffected. In this scenario, the mother was unaffected. The scenario here, the mother is affected. So now let's see how that works. Now the father has got um, a Y chromosome, which is good, and he's got also an X chromosome that is good. So the father has got good chromosomes from his parents. His mom passed a good X chromosome, and his father passed a good Y chromosome. So the father is happy. He is unaffected. Okay, the mother um, is having a problem here. Here, the mother was all okay, no affected. Everything was okay. She inherited both good copies of the gene. Now here is a problem. It's got one bad copy and one good copy. So the mother is a carrier. It's not affecting her, but she's carrying whatever disease this could cause when it meets up with another bad copy. So when they have a child, this is the scenario that could happen. This is what is called an X-link. Like, okay, if they have an unaffected daughter, the daughter should be having XX chromosome because it's a female. Female should have XX. So he's taking the X. The father must have passed the X to the mother, and the mother have one good X and one bad X. So it will take the good X from the mother and drop the bad X. So the baby will end up having two good X chromosomes, and the baby is unaffected. Now, if they have a son, look at what could happen here as well. The son is an XY chromosome, and the Y of the father has to go to the mother. The Y is not affected, and it will take one of the X chromosomes of the mother, and the mother also have one good and one bad. So the son will take the good chromosome from the mother and a good chromosome from the father, and the son is also unaffected. Now, in this scenario, if they have a daughter, who should have XX, so the father would pass one good X, and the mother would pass also one X. So the X that the mother is passing here could be one of these ones, could be an affected copy, and because he's already get a good copy from the father and taking a bad copy from the mother, then that daughter is affected. But it's a carrier. It's not showing any symptoms, but it's carrying whatever disease that could be. So if she meets up with a partner who is also unaffected but is a carrier, then their child will have inherited both bad copies. Now, they have a son. The son is an XY chromosome. It's a male. So it will take the Y, which is from the father, is good. And it will take an X from the mother. It will take the bad X from the mother. And then that son could be affected. So this is how X-linked dominant inheritance can happen. If the X-link is passed on from the mother, so the dad could inherit an X from the parents, and then when he got married and finds a woman with two X's, then the mixing of the genes can take place there, and this is what the results could be. Now, X-link also could cause diseases that are very fatal, like Menkes disease. The Menkes disease is a disease that develops in a child very early in age, and it causes very serious, devastating effects to the child. It usually appears before 3 months. You can start to see the effects on this baby. And the general outcome is that the baby deteriorates towards 5 to 6 years, and then the baby will pass away. And this can cause many serious problems that parents with children that passed away all the time might have this X-linked dominant inheritance disease called Menkes disease. Right. So now we're going to talk about quickly um, exogenous mutations. What we were talking about was all endogenous. It was things happening inside the gene. Now let's talk about things happening outside the gene. Outside the gene here, you can see electromagnetic spectrum is here, which is full of radiations. The radio waves we capture from here at this wavelength, microwaves operate at this wavelength, infrared remote controls operate at this wavelength. Our visible eyes can see only in this wavelength, this small narrow band. You have ultraviolet radiations here, X-rays at the hospital operates here, and you have gamma rays in that region. Gamma rays are very terrible. They have a very high frequency and they can penetrate all solid substances. And this is what happens in nuclear radiations. Right. Okay, so you can see these are warnings that they post in hospitals that it says, attention patients, if you are pregnant or think you may be pregnant, please inform the nurse radiation is in use here. So because radiation damages the DNA and is an exogenous damage, is something that is coming from outside your environment, it is outside your body, from your environment, getting into your body, and it is very bad in DNA molecules. It can damage the DNA and it will change the DNA, and mutation can happen, and you will have a mutated child. And these notices are posted in hospitals, and we have to be aware of them. If you are pregnant, you have to inform the nurse or you have to move away from these areas. Now, this is called radioactive mutation that I have shown in the example lecture that I have put up. This happened in Chernobyl when the nuclear catastrophe happened there. The nuclear power station exploded and contaminated the whole area with radioactive radiations. So the animals that lived in that area were affected due to the radiation. And when this animal was in its mother's womb, there was changes in the DNA that gave about this mutation. So it became two-headed. Now this is also another one. It's also a mutation in animals, and this was also in the Chernobyl area. So this is what radioactive mutation can cause. Now here you can see it in humans. This child has got its fingers um, all messed up due to wrong DNA sequencing. And this child has got multiple faces. You can pause the video and read about them. And here you can have a baby with 4 legs as well. It's all due to the genes. What the genes are supposed to make, the mutation have changed the gene, and the gene now is making something different. This is a thalidomide babies. They call them the thalidomide babies because what happened in the UK, there was a period of time that um, pregnant women were given a thalidomide medication, and it was to help them with their morning sickness. But during that period of time, over 2 years or so, babies that were getting born, were disfigured like these. And scientists were wondering what could have been the cause. So they started their investigations. They go through the medical records of women who were pregnant, and they found that they were all having this thalidomide medication, which was for their morning sickness. and it was the thalidomide medication, which is a chemical that also is an exogenous. It is taken from the external environment, from your body, and then you put it in your body, and it interacts with your DNA, and it changes the DNA. And the mutation happens, and you produce such a child like that. So when they knew that, thalidomide was banned throughout in Europe. I don't know whether in Africa as well, but beware of that. Thalidomide should not be consumed. Now, we come to albinism, mutations that can cause albinism. That albinism, we all know what it is. You become an albino. It's caused by the tyrosinase protein that causes the pigment in our bodies. It's being located in chromosome number 11, and it is in location Q14 and Q21 region of that protein, of that DNA, and that gene spans about 65 kilobases, 65,000 base pairs in that region. And that region, if it is corrupted, then it cannot produce the pigment that your body needs, and then you become a mutant albino. So you don't have the pigmentation that your body and your hair needs to make you what you're supposed to be. Also, this is an inherited disease. Your parents must have to have been carriers, and then they marry together, and you inherit bad copies, two bad copies, so the copies cannot make anything good. So that is why you become an albino. Albinism also happen in animals. You can see horses, you can see a turtle here. These are all black turtles, but this turtle is albino. It becomes a white turtle. That's an albino human. It's very rare for you to see albino animals because animals live in the wild, and they are hunted by other animals to eat them, because in the wild is like a predator and prey. Predators are predating on their prey to eat them, to have something as food. So if you lost your camouflage and you cannot hide anymore, you will be visible, you will be seen, and therefore you will be easy to pick out, and you will be eaten. So that is why it is very rare for you to see albino animals in the wild, because they are vulnerable, they cannot hide, they are too visible, and they are picked up easily and eaten. Now, the next thing is achondroplasia. Achondroplasia is another genetic disorder where you become a short limb dwarfism. It's the short limb dwarfism that the people who are very short, and the problem they have is from the chromosome number 4. The FGFR number 3 gene on chromosome number 4 is what is affected in them. 1 in 25,000 individuals of all ethnic groups have this achondroplasia. And you can see the achondroplasia here in different people, different animals, as we are all having the same genes, animals, plants, and humans. Plants do not have achondroplasia because they don't have any bone structure in them. But all living things with bone structure can suffer from achondroplasia. And now that ends our discussion. I to recap, I just want you to go back, rewind this video, have a look at the chromosomal sets that I showed you at the beginning, try and understand the different type of mutations that can happen and the diseases that they can cause. Try to understand that whatever your child is, you are the parents that give it to that child. It's not anything accidental. It's not anything that is from spiritual. It's not anything that came from nowhere. It is from you. You gave it to the child. So be very careful about who your partner is, about what you do. You understand? And I thank you very much for listening. And I hope to come again in this channel with another lesson. And that lesson now is going to be sex determination. How do we have males and females in animals, humans, and in plants? Thank you very much.